Hi class, this is your instructor, Skylar Huff, and welcome to Energy and Metabolism. Life can be viewed as a constant flow of energy, channeled by organisms to do work through living. Each of these significant properties by which we define life, order, growth, reproduction, responsiveness, and internal regulation requires a constant supply of energy. Energy that the lion extracts from its meal of a giraffe will be used to run its cells, power its roar, fuel its running, and build a bigger lion. Deprived of an energy source, life stops. Therefore, a comprehensive study of life would be impossible without discussing bioenergetics, the analysis of how energy powers the activities of living systems. So let's focus on energy, what it is, how it changes during chemical reactions. So we begin with the flow of energy in living systems, laws of thermodynamics. Thirdly, we'll get to energy currency called ATP, and then end the chapter with enzymes known as biological catalysts, and then metabolism. Thermodynamics is the branch of chemistry concerned with energy changes. Cells are governed by the laws of physics and chemistry, so we must understand these laws in order to understand how cells function. Energy is defined as the capacity to do work. We think of energy existing in two states, kinetic and potential. Kinetic energy is energy of motion. Moving objects perform work by causing other matter to move. Potential energy is stored energy. Objects that are not actively moving but have the capacity to do so possess potential energy. A boulder perched on a hilltop has gravitational potential energy. As it begins to roll down the hill, some of its potential energy is converted into kinetic energy. Much of the work of that living organism carry out involves transforming potential energy into kinetic energy. And energy can take on many forms: mechanical energy, heat, sound, electric current, light, or radioactivity. Because it can exist in so many forms, energy can be measured in many ways. Heat is the most convenient way of measuring energy because all other forms of energy can be converted into heat. In fact, the term thermodynamics means heat changes. The unit of heat most commonly employed in biology is the kilocalorie. 1 kilocalorie is equal to 1,000 calories. 1 calorie is the heat required to raise the temperature of 1 gram of water 1 degree Celsius. Another energy unit often used in physics is the joule. 1 joule equals 0.239 calories. So you're seeing figure 6.1 depicting in 6.1a potential energy and in 6.1b kinetic energy. Energy flows in most ecosystems from the sun. It is estimated that sunlight provides the Earth with more than 1.3 times 10 to the 24 calories per year, or 4 times 10 to the 16th calories per second. Plants, algae, and certain kinds of bacteria capture a fraction of this energy through photosynthesis. There are also ecosystems where bacteria extract energy from the oxidation of inorganic compounds. As we will see, these initial sources of energy literally power the energetic needs of all living systems. In chemical reactions, breaking chemical bonds requires energy, and forming covalent bonds releases energy. If a reaction forms a more ordered, higher energy state, as photosynthesis does, an input of energy is required. In photosynthesis, energy is absorbed from sunlight, is used to combine small molecules into more complex ones. In the process, energy from sunlight is stored as potential energy in the structure of the sugar molecules. During a chemical reaction, the energy stored in chemical bonds may be used to make new bonds. One important class of reactions involves the transfer of one or more electrons from one atom or molecule to another. The atom or molecule that loses an electron is said to be oxidized, and the process is called oxidation. The atom or molecule that gains the electron is said to be reduced. The process is called reduction. The reduced form of a molecule has a higher level of potential energy than the oxidized form. Oxidation and reduction always take place together because every electron that is lost by one atom through oxidation is gained by another atom through reduction. Therefore, chemical reactions of this sort are called oxidation reduction or redox reactions. Redox reactions transfer energetic electrons when bonds are made or broken. And with these electrons, the potential energy that electrons hold. It is for this reason that the reduced form of a molecule has a higher level of energy than the oxidized form. Oxidation and reduction reactions play a key role in the flow of energy through biological systems. In the next two chapters, you will learn the details of how organisms derive energy from the oxidation of organic compounds via respiration, as well as the energy in sunlight via photosynthesis. Thermodynamics is a branch of chemistry concerned with energy changes. Cells are governed by the laws of physics and chemistry, so we must understand these laws in order to understand how cells function. All activities of living organisms, growing, running, thinking, singing, or even when you read, involves changes in energy. Two universal laws, which we call the laws of thermodynamics, govern all energy changes in the universe, from the nuclear reactions that occur in the sun to your picking up your textbook. The first law of thermodynamics concerns the amount of energy in the universe. Energy cannot be created or destroyed. It can only change from one form to another, from potential to kinetic, for example. The total amount of energy in the universe remains constant. The lion eating the giraffe at the beginning of this chapter is acquiring energy rather than creating new energy. The lion is transferring some of the potential energy stored in the giraffe's tissues to its own body, just as the giraffe obtained potential energy stored in the plants it ate while it was alive, and those plants captured energy from sunlight. Within any living organism, chemical potential energy stored in some molecules can be shifted to other molecules and stored in different chemical bonds. It can also be converted into other forms such as kinetic energy, light, or electricity. During each conversion, some of the energy dissipates into the environment as heat, which is a measure of the random motion of molecules. Energy continuously flows through the biological world in one direction, with new energy from the sun constantly entering the system to replace the energy dissipated as heat. Heat can be harnessed to do work only when there is a heat gradient, that is, a temperature difference between two areas. Cells are too small to maintain significant internal temperature differences, so heat energy is incapable of doing the work of cells. Instead, cells must rely on chemical reactions for energy. Although the total amount of energy in the universe remains constant, the energy available to do work decreases as more of it's progressively lost as heat. The second law of thermodynamics addresses the efficiency of energy transformations. Energy cannot be transformed from one form to another with 100% efficiency. Some energy is always unavailable. This unavailable energy manifests as an increase in the random motion of molecules, an increase in the number of energy states available to atoms, or an increased dispersal of energy in the system. This is often characterized as an increase in the randomness or disorder of the system. We measure this as an increase in entropy, symbolized by the letter S in the equations in the next section we're going to. Put simply, disorder is more likely than order. For example, it is more likely that a column of bricks will fall over than for that pile of bricks will spontaneously form a column. Energy transformations proceed spontaneously to convert matter from a more ordered or less stable form to a less ordered but more stable form. Consider a glass of water in a room at 25 degrees Celsius. The water molecules in the ice form an ordered, regular array, while the water molecules in the liquid are more disordered. There are far fewer ways that molecules can be arranged in the crystal than in the liquid water. As the ice absorbs the heat from the surroundings, the hydrogen bonds holding the crystals together are broken and the water molecules become less ordered, higher entropy. When all the ice has melted, the entropy of the water will be maximal for the system. So you can order the images in figure 6.3 in your text based on experience, but the underlying reason is the second law. For this reason, it is sometimes called times arrow. And there you have figure 6.3, times arrow. It takes energy to break chemical bonds that hold atoms in a molecule together. Heat energy, because it increases atomic motion, makes it easier for atoms to pull apart. For this reason, both chemical bonding and heat have a significant thermodynamic influence on a molecule, the former reducing disorder and the latter increasing it. The net effect, the amount of energy actually available to break and subsequently form other chemical bonds, is called the free energy of that molecule. We define free energy as energy available to do work at constant temperature in a system. For conditions of constant temperature, pressure, and volume, we call this Gibbs free energy, symbolized by G. So Gibbs free energy is equal to the total energy in a molecule's chemical bonds, called enthalpy and designated as H, minus a term, Ts, that expresses the amount of disorder in a system, where S is entropy and T is the absolute temperature in degrees Kelvin. So here is the equation. Chemical reactions may break some bonds in the reactants and form new bonds in the products. Consequently, reactions can produce changes in free energy. When a chemical reaction occurs under conditions of constant temperature, pressure, and volume, as do most biological reactions, the change symbol, shown as the Greek letter delta, in free energy is simply the change in Gibbs free energy equals the change in enthalpy minus the change in entropy times temperature. So again, the change in Gibbs free energy is the change in enthalpy minus temperature times entropy. And that's that change in entropy. We can use the change in free energy to predict whether a particular chemical reaction is spontaneous or not. The change in free energy is calculated as the energy of products minus the energy of reactants. So if the products have more free energy than the reactants, delta G is positive. Such reactions are not spontaneous because they require an input of energy. This can be either because the bond energy is higher or the disorder in the system is lower. We call this an endergonic reaction. A plot of free energy for reactants and products shows this graphically in figure 6.4a. An endergonic reaction is uphill from reactants to products and not spontaneous. Conversely, if the products have less free energy than the reactants, delta G is negative and the reaction will proceed spontaneously. The negative delta G can be because the bond energy is lower or the disorder is higher or both. These reactions release energy and are called exergonic reactions, and the plot of free energy for the reaction is now downhill from reactants to products, and the reaction will be spontaneous. Note that spontaneous does not mean the same thing as instantaneous. A spontaneous reaction may proceed very slowly. Because chemical reactions are reversible, a reaction that is exergonic in the forward direction will be endergonic in the reverse direction. If all chemical reactions that release free energy tend to occur spontaneously, why haven't all such reactions already occurred? Consider the gasoline tank of your vehicle. The oxidation of the hydrocarbons in gasoline is an exergonic reaction, but your tank does not spontaneously explode. One reason is that most reactions require an input of energy to get started. In the case of your vehicle, this input of the electrical sparks in the engine's cylinders, producing a controlled explosion. Before new chemical bonds can form, even bonds that contain less energy, existing bonds must first be broken, and that requires energy input. The extra energy needed to destabilize existing chemical bonds and initiate a chemical reaction is called activation energy. You may see figure 6.5. The rate of an exergonic reaction depends on the activation energy required for the reaction to begin. Reactions with larger activation energies tend to proceed more slowly because fewer molecules will collide with sufficient energy to get over the initial energy hurdle. The rate of reactions can be increased in two ways. One, by increasing the energy of reacting molecules, or two, by lowering activation energy. Chemists often drive important industrial reactions by increasing the energy of the reacting molecules, which is frequently accomplished by simply heating up the reactants. The other strategy is to use a catalyst to lower the activation energy. So here is figure 6.5 showing you activation energy and catalysts. Stressing particular chemical bonds can make them easier to break. The process of influencing chemical bonds in a way that lowers the activation energy needed to initiate the reaction is called catalysts. And the substances that accomplish these are known as catalysts. Catalysts exert their action by affecting an intermediate stage in the reaction, the transition state. The energy needed to reach this transition state is the activation energy. Catalysts stabilize this transition state, thus lowering the activation energy. Catalysts cannot violate the basic laws of thermodynamics. They cannot make an endergonic reaction proceed spontaneously. By reducing the activation energy, a catalyst accelerates both the forward and the reverse reactions by exactly the same amount. Therefore, a catalyst does not alter the proportion of a reactant ultimately converted into a product. To understand this, imagine bowling a ball resting in a shallow depression on the side of a hill. Only a narrow rim of dirt below the ball prevents it from rolling down the hill. Now imagine digging away that rim of dirt. If you do remove enough dirt from below the ball, it will start to roll down the hill, but removing dirt from below the ball will never cause the ball to roll up the hill. Removing that lip of dirt simply allows the ball to move freely. Gravity determines the direction it then travels. Similarly, the direction in which a chemical reaction proceeds is determined solely by the difference in the free energy between reactants and products. Like digging away the soil below the bowling ball and hill, catalysts reduce the energy barrier that is preventing the reaction from proceeding. Only exergonic reactions can proceed spontaneously, and catalysts cannot change that. What catalysts can do is make the reaction proceed much faster. In living systems, enzymes act as catalysts. And you see here in blue the amount of activation energy needed for this uncatalyzed reaction. You also see how long it takes that uncatalyzed reaction to proceed. In red is that enzyme catalyzed reaction. You see that lowered activation energy, or that energy of activation needed, and you also see how quickly that reaction proceeds. Adenosine triphosphate was both a precursor for RNA and also the energy currency of the cell. We call ATP the energy currency because the energy released by ATP hydrolysis can power most endergonic reactions. The process of energy metabolism in cells is complex, as we shall see in the next two chapters. So to provide energy when needed, cells use energy released by the exergonic reactions of energy metabolism to synthesize an intermediate that can later be degraded to release energy on demand. The molecule that has evolved to fulfill this role in cells is ATP. ATP is composed of three small components. Please see figure 6.6. The first component is a 5-carbon sugar, ribose, which serves as the framework to which the other two subunits are attached. The second component is adenine, an organic molecule composed of 2 carbon nitrogen rings. Each of the nitrogen atoms in the ring has an unshared pair of electrons and weakly attracts hydrogen ions, making adenine chemically a weak base. The third and final component of ATP is a chain of 3 phosphates, thus adenosine triphosphate. The key to how ATP stores energy lies in its triphosphate group. Phosphate groups are highly negatively charged, and thus they strongly repel one another. This electrostatic repulsion makes the covalent bonds joining the phosphates unstable. The molecule is also often referred to as a coiled spring, with the phosphates straining away from one another. The unstable bonds holding the phosphates together in the ATP molecule have a low activation energy and are easily broken by hydrolysis. This reaction is exergonic, releasing a considerable amount of energy. In other words, the hydrolysis of ATP is a negative delta G, and the energy it releases can be used to perform work. For this reason, we often refer to these high energy bonds, referring to this release of energy, not to the actual bond energy. In most reactions involving ATP, only the outermost high-energy phosphate bond is hydrolyzed, clipping off the phosphate group at the end. What happens when ATP becomes adenosine diphosphate plus an inorganic phosphate, and equal to 7.3 kilocalories per mole is released under standard conditions. This reaction can also transfer the terminal phosphate to another molecule when it's hydrolyzed. It will release inorganic phosphate. Both of the terminal phosphates can be hydrolyzed to release energy, leaving adenosine monophosphate, but the third phosphate is not attached to a high-energy bond with only one phosphate group. AMP has no other phosphates to provide the electrostatic repulsion that makes the bonds holding the two terminal phosphate groups high-energy bonds. This is ATP. Cells use ATP to drive endergonic processes. Remember that the endergonic reactions do not proceed spontaneously because their products possess more free energy than their reactants. Delta G for an endergonic reaction is positive. If the ATP hydrolysis releases more energy than the other reaction consumes, then coupling the two reactions will produce an overall delta G for the coupled reactions that is negative. That is, the energy released by the hydrolysis of ATP can supply the energy needed for the endergonic reaction. Because most endergonic reactions in cells require less energy than is released by ATP hydrolysis, ATP can provide most of the energy a cell needs. ATP also powers other cellular functions such as generating force in muscles or creating concentration gradients for important ions, both processes that require energy. The same feature that makes ATP an effective energy donor, the inability of its phosphate bonds, make it a poor long-term energy storage molecule. Fats and carbohydrates serve that function better. Cells make and use ATP cyclically. Cells use exergonic reactions to provide energy to synthesize ATP from ADP plus that inorganic phosphate. They then use the hydrolysis of ATP to provide energy to drive endergonic processes. So most cells do not maintain large stockpiles of ATP. Instead, typically they have only a few seconds supply of ATP at any given time, and they continue to produce more from ADP and inorganic phosphate. So it is estimated that even sedentary individuals turn over an amount of ATP in one day roughly equal to his or her body weight. This statistic makes clear the importance of ATP synthesis. That's an amazing statistic there. So here's that cycle. Chemical reactions in living organisms are regulated by controlling the points at which catalysts takes place. Life itself, therefore, can be seen as regulated catalysts, being regulated by catalysts. This is possible because the catalysts in living systems, enzymes, are also highly specific. Most enzymes are proteins, although RNA molecules can also have catalytic activity. The unique three-dimensional shape of an enzyme enables it to stabilize a temporary association between substrates, the molecules that will undergo the reaction. By bringing two substrates together in the correct orientation or stressing particular chemical bonds of a substrate, an enzyme lowers the activation energy required for bonds to form. The reaction thus proceeds more quickly than it would without the enzyme. The enzyme itself is not changed or consumed by the reaction, so only a small amount of the enzyme is needed, and it can be used repeatedly. As an example of how an enzyme works, let's consider the reaction of carbon dioxide and water to form carbonic acid. This important enzyme catalyzed reaction occurs in vertebrate red blood cells. This reaction may proceed in either direction, from CO2 plus water to carbonic acid, or from carbonic acid to CO2 and water. So it may proceed in either direction because it has a large activation energy, and the reaction is very slow in the absence of an enzyme. Perhaps 200 molecules of carbonic acid form in an hour in a cell in the absence of any enzyme. Reactions that proceed this slowly are of little use to a cell. Vertebrate red blood cells overcome this problem by employing an enzyme within their cytoplasm called carbonic anhydrase. Under the same conditions, but in the presence of carbonic anhydrase, an estimated 600,000 molecules of carbonic acid form every second. Thus, the enzyme increases the reaction rate by more than 1 million times. Thousands of different kinds of enzymes are known, each catalyzing one or a few specific chemical reactions. By facilitating particular chemical reactions, the enzymes in a cell determine the course of metabolism, the collection of all chemical reactions in that cell. Different types of cells contain different sets of enzymes. and this difference contributes to structural and functional variations among cell types. For example, the chemical reactions taking place within a red blood cell differ from those occurring within a nerve cell, in part because of different cell types contain different arrays of enzymes. Most enzymes are globular proteins with one or more pockets or clefts called active sites, shown there at top left. Substrates bind to the enzyme at active sites, forming an enzyme-substrate complex, shown here at figure 6.8b. For catalysis to occur within the complex, a substrate molecule must fit precisely into an active site. When that happens, amino acid side groups of the enzyme end up very close to certain bonds of the substrate. These side groups interact chemically with the substrate, usually stressing or distorting a particular bond, and consequently lowering the activation energy needed to break the bond. After the bonds of the substrates are broken or new bonds are formed, the substrates have been converted into products. These products then dissociate from the enzyme, leaving the enzyme ready to bind its next substrate and begin the cycle again. Proteins are not rigid. The binding of a substrate induces the enzyme to adjust its shape slightly, leading to a better or induced fit between the enzyme and substrate. This interaction may also facilitate the binding of other substrates. In such cases, one substrate activates the enzyme to receive other substrates. Although many enzymes are suspended in the cytoplasm of cells, not attached to any structure, other enzymes function as integral parts of cell membranes and organelles. Enzymes also form associations with multi-enzyme complexes to carry out reaction sequences, and it is now clear that some enzymes may contain catalytic RNA rather than only being protein. So in figure 6.10 shown for you here, this is the catalytic cycle of an enzyme. Substrate sucrose consists of glucose and fructose bound together. The substrate binds to the active site of the enzyme, forming that enzyme substrate complex. The enzyme here is called sucrase. Substrate is sucrose. The binding of the substrate and the enzyme places stress on the glucose-fructose bond. The bond breaks. The products are released, being glucose and fructose. Products are released, and enzyme is free to bind with other substrates, i.e. more sucrose, to form more glucose and fructose. Thanks a lot, sucrase. Often several enzymes catalyzing different steps of a sequence of reactions are associated with one another in non-covalently bonded assemblies called multi-enzyme complexes. The bacterial pyruvate dehydrogenase multi-enzyme complex, shown in figure 6.11, contains enzymes that carry out 3 sequential reactions in oxidative metabolism. Each complex has multiple copies of each of the 3 enzymes, 60 protein subunits in all. The many subunits work together to form a molecular machine that performs multiple functions. Multi-enzyme complexes offer the following significant advantages in catalytic efficiency. The rate of enzyme reaction is limited by how often enzymes collides with the substrates. So if a series of sequential reactions occurs a multi-enzyme complex, the product of one reaction can be delivered to the next enzyme without releasing it to diffuse away. 2. Because reacting substrate does not leave the complex while it goes with a series of reactions, unwanted side reactions are prevented. And thirdly, all of the reactions take place within the multi-enzyme complex can be controlled as a unit. The original enzymes discovered and studied were all proteins. For many years, we assumed that all enzymes were proteins until Thomas R. Seck and colleagues at the University of Colorado reported in 1981 an RNA splicing reaction that did not require a protein. Around the same time, Sidney Altman and Norman Pace were studying the enzyme RNase P and found that this enzyme was composed of both protein and RNA, and further, that the RNA was the catalytic molecule. Like protein enzymes, these RNA catalysts, which are called ribozymes, greatly accelerate the rate of a particular biochemical reaction and show extraordinary substrate specificity. Research has revealed at least two sorts of ribozymes. Some ribozymes have folded structures and catalyze reactions on themselves, called intramolecular catalysis. Other ribozymes act on molecules without being changed themselves, a process called intermolecular catalysis. The rate of enzyme catalyzed reaction is affected by concentration of both the substrate and the enzyme that works on it. In addition, any chemical or physical factor that alters the enzyme's three-dimensional shape, such as temperature, pH, and the binding of regulatory molecules, can affect the enzyme's ability to catalyze the reaction. Increasing the temperature of an uncatalyzed reaction increases its rates because additional heat increases random molecular movement. This increases the fraction of molecules whose kinetic energy is greater than the activation energy for the reaction. The rate of an enzyme catalyzed reaction also increases with temperature, but only up to a certain point called the optimum temperature. Below this temperature, the hydrogen bonds and hydrophobic interactions that determine the enzyme's shape are not flexible enough to permit the induced fit that is optimum for catalysis. Above the optimum temperature, these forces are too weak to maintain the enzyme's shape against the increased random movement of the atoms in the enzyme, and at higher temperatures, the enzyme denatures. Most human enzymes have an optimum temperature between 35 degrees Celsius and 40 degrees Celsius, a range that includes normal body temperature. Prokaryotes that live in hot springs have more stable enzymes, that is, enzymes held together more strongly. So the optimum temperature for those enzymes can be 70 degrees Celsius or higher. In each case, the optimal temperature for the enzyme corresponds to the normal temperature usually encountered in the body or environment depending on the type of organism. Ionic interactions between oppositely charged amino acid residues such as glutamic acid and lysine also hold enzymes together. These interactions are sensitive to the hydrogen ion concentration of the fluid in which the enzyme is dissolved, because changing that concentration shifts the balance between positively and negatively charged amino acid residues. For this reason, most enzymes have an optimum pH that usually ranges from pH 6 to 8. Enzymes able to function in very acidic environments are proteins that maintain their three-dimensional shape even in the presence of high hydrogen ion concentrations, and the enzyme pepsin, for example, digests proteins in the stomach at pH 2, a very acidic level. So this is now figure 6.12, showing you enzyme sensitivity to the environment. So at the top is temperature. In blue, optimal temperature for human enzyme. In red, optimal temperature for prokaryotes found in hot springs. And at figure 6.12b, you have the pH optimum for pepsin, roughly 2.5. And then you have the pH optimum for trypsin, roughly 6.5. So clearly trypsin will be found in the small intestine, whereas pepsin will be found in the stomach. Enzyme activity is also sensitive to the presence of specific substances that can bind to the enzyme and cause changes in its shape. Through these substances, a cell is able to regulate which of its enzymes are active and which are inactive at a particular time. This ability allows the cell to increase its efficiency and to control changes in its characteristics during development. A substance that binds to an enzyme and decreases its activity is called an inhibitor. Very often, the end product of a biochemical pathway acts as an inhibitor of an early reaction in the pathway, a process called feedback inhibition. Enzyme inhibition occurs in two ways. Competitive inhibitors compete with the substrate for the same active site, occupying the active site and thus preventing substances from binding. Non-competitive inhibitors bind to the enzyme in a location other than the active site, changing the shape of the enzyme and making it unable to bind to the substrate. Many enzymes can exist in either an active or inactive conformation. Such enzymes are called allosteric enzymes. Most non-competitive inhibitors bind to a specific portion of an enzyme called the allosteric site. These sites serve as chemical on-off switches, the binding of a substance to a site that can switch off the enzyme between its active and inactive conformations. A substance that binds to an allosteric site and reduces enzyme activity is called an allosteric inhibitor. This kind of control is also used to activate enzymes. An allosteric activator binds to allosteric sites to keep enzymes in its active configuration, thereby increasing enzyme activity. So in figure 613A is a competitive inhibitor. In figure 613B, you have non-competitive inhibition, and you also have that allosteric inhibitor on that allosteric site. When a cofactor is a non-protein organic molecule, it is called a coenzyme. Many of the small organic molecules essential in our diets, we call vitamins, function as coenzymes. For example, the B vitamins, B6 and B12, both function as coenzymes for a number of different enzymes. Modified nucleotides are also used as coenzymes. In numerous oxidation reduction reactions that are catalyzed by enzymes, the electrons pass in pairs from active site of the enzyme to a coenzyme that serves as the electron acceptor. The coenzyme then transfers the electron to a different enzyme, which releases them and the energy they bear to the substrates in another reaction. Often the electrons combine with protons to form hydrogen atoms. In this way, coenzymes shuttle energy in the form of hydrogen atoms from one enzyme to another in a cell. Living chemistry, the total of all chemical reactions carried out by an organism, is called metabolism. Those chemical reactions that expend energy to build up molecules are called anabolic reactions or anabolism. Reactions that harvest energy by breaking down molecules are called catabolic reactions or catabolism. We have already seen examples of this in Chapter 3, where we saw that biopolymers are built by anabolism, known as those dehydration reactions, and degraded or catabolized by hydrolysis reactions. Organisms contain thousands of different kinds of enzymes that catalyze a bewildering variety of reactions. Many of these reactions in a cell occur in sequences called biochemical pathways. In such pathways, a product of one reaction becomes the substrate for the next. Biochemical pathways are the organizational units of metabolism, the elements an organism controls to achieve coherent metabolic activity. Many sequential enzyme steps in biochemical pathways take place in specific compartments in the cell. For example, the steps of the citric acid cycle occurs in a matrix inside the mitochondria in eukaryotes. We'll be there in the next chapter. By determining where many of the enzymes that catalyze these steps are located, we can map out a model of the metabolic process in the cell. The majority of these biochemical pathways are common in all cells, which implies that a basic cell metabolism evolved early in the history of life on this planet. This has left modern cells with a metabolism where carbon and energy pathways converge to produce 12 key intermediates that are the precursors for biosynthetic pathways. So this is a biochemical pathway, and the original substrate is acted on by enzyme 1. changing the substrate into a new intermediate, substrate A, recognized as a substrate by enzyme 2. Each enzyme in a pathway acts as a product of the previous stage, and these enzymes may be either soluble or arranged in a membrane as shown. For a cell to operate efficiently, the biochemical pathways that make up its metabolism must be coordinated. Not only is it unnecessary to synthesize a compound when it's not enough available, not only is it unnecessary to synthesize a compound when enough is available, but it is a waste of energy and raw materials that could be used elsewhere in the cell. It is advantageous for the biochemical pathways to be active only when their products are needed. The regulation of simple biochemical pathways often depends on an elegant feedback mechanism. The end product of the pathways binds to an allosteric site on the enzyme that catalyzes the first reaction in the pathway. This mode of regulation is called feedback inhibition. So in the hypothetical biochemical pathway shown here with 3 enzymes, the initial substrate is converted into the end product via 2 intermediate substrates A and B. With no feedback inhibition, the pathway keeps churning out end product as long as the initial substrate is available. With feedback inhibition, as the end product builds up, it can bind to an allosteric site in the enzyme 1 to shut off the pathway. So in this chapter, we have reviewed the basics of energy and its transformations as carried out in living systems. Chemical bonds are the primary location of energy storage and release, and cells have developed elegant methods for making and breaking chemical bonds to create molecules in need. Enzymes facilitate these reactions by serving as catalysts. So in the next 2 chapters, you will learn the details of mechanisms by which organisms harvest, store, and utilize energy. This has been your instructor.